Hi, my name is Julieta Trejo, and I'm under the mentorship of Dr. Yuan Bo Peng for the Department of Psychology. Today I'm going to be talking about the local field potential change during euthanasia, Part 1, in anesthetized rats. This project was done in collaboration with Caitlin, Melissa, and Zen, who are a part of the Freely Moving group, and also the group that compares both studies. So near-death experience, also known as NDE, is a fascinating topic that has raised many unanswered questions. After surviving a life-threatening crisis, many individuals report encountering a near-death experience. Near-death studies explain this experience as an out-of-body experience of consciousness traveling to a different dimension. A similar, perhaps closely related phenomenon is terminal lucidity. Terminal lucidity is a term that refers to the short burst of mental clarity or mental improvement that may happen soon before the end of life. Specifically, terminal lucidity has shown prevalence in individuals with mental disorders such as Alzheimer's, schizophrenia, dementia, etc. What causes terminal lucidity is still unknown. In the present experiment, from the end of another experiment when animals were euthanized, we demonstrate the local field potential changes in rats from intracranially implanted electrodes. These electrodes were implanted into 4 regions of the rat's brain, which were the anterior cingulate cortex, also known as ACC, the thalamus, also known as PO, trigeminal ganglion, also known as TG, and the primary visual cortex, also known as V1M. The local field potential can be subdivided into 5 frequency bands of delta, theta, alpha, beta, and gamma. So for the methodology of the study, we used 44 adult male rats, each weighing between 300 grams and 400 grams. There were 2 groups of freely moving, with the freely moving group had 18 rats and the anesthetized had 21 rats. For the surgical procedure, 4 electrodes were implanted in the ACC, right thalamus, left TG, and right V1M, along with 2 reference screws. The screws were connected to the wireless module that allows the electrodes to transmit data to the recording software. And if you look here to the bottom left, you can see that the little white cylinders are actually the 4 electrodes. And then we have 2 little screws right here with a red wire poking out, and this red wire is where we connect our recording device. So after all of this is set up, the animal is then injected with nitroglycerin, also known as NTG, and recorded for 4.5 hours. After the NTG, the animal then undergoes the euthanasia process, and for this process, we recorded 5 minutes of baseline activity, and then the CO2 is turned on. After the CO2 is turned on, an additional 30 minutes is recorded. So here for the results, if you look up here, this sequence of photos shown above is one of the programs that we used called Spike2. Spike2 measures the amplitude of different channels and regions of the brain. Here, ACC, PO, TG, and V1M areas are measured. Now, these figures below, in the graphs, basically the study revealed a general decrease of power in all 5 frequency bands approximately 2 minutes after CO2 was turned on. And as you can see here, this is actually normalized data. This is the 10-second side versus the 1 minute for each brain, ACC, TG, PO, and V1M. So as you can see, there is a general decrease of power, and this is a trend in basically all of these graphs. So additionally, after turning on CO2, a significant drop was found between time 7 to 35. So finally, in conclusion, the local field potential power changes in the anesthetized group establishes the basis for further study of the dynamic euthanasia process in the brain. Thank you so much.